Okay, good morning, everyone. How are you today? I hope you all are safe and well. Yesterday, on the previous lesson, we studied in biology that about Darwin's theory of evolution. And we said that members of species have different characteristics. We all have different characteristics and many different useful and non-useful characteristics. And also, these make some individuals more likely to survive than others. The survivors reproduce and pass their useful characteristics to the next generation. Over many generations, these characteristics become more common and species changes. So because they are passing their useful genes to their offsprings, so over years, this will become more common. Today we will study about moving genes. How can genes move across generations and how can they be adapted to their environments and how can they improve their bodies or their living cells. So please grab your notebooks, your pencils, and write these notes with me. Moving genes, adding genes. Adding new genes to the cells is called genetic engineering. So we all heard about something called genetic engineering, but what is this genetic engineering exactly? When scientists want to breed some animals or organisms, sometimes they have to add new genes to the cells. So when they add these useful ones to get useful characteristics or strong characteristics, they have to add genes. This is called genetic engineering. They're using this more than the, even like plus the selective breeding. Sometimes they need to have the selective breeding, but genetic engineering successful also. Genetic engineering is tricky to carry out. It's hard. It's not easy to extract a gene and inject it or add it to the cells because this is like very sensitive. It's not easy, but it has many advantages because this can give you more useful characteristics, can improve the crops, can improve the health of the animals, can get them new characteristics or features. It can be used to add genes that aren't usually found in that species. And it can produce animals with new characteristics more likely. I'm going to show you a video about this. How can they use genetic engineering to add or use genetic engineering to add new characteristics in animals that they don't have exactly. Some of them, they can have glowing in their bodies, glowing cats, glowing rats. They are extracting a gene from jellyfish because we all know that we have some kinds of jellyfish that can glow. So they are extracting that genes from them and injected or added this gene to the cells of a rat or of any animal that they wanted to glow. But it cannot just glow like this. It will glow under ultraviolet light, okay? So let's see together the genetic engineering and how this can affect the rats. This is a mouse, ordinary mouse, but it has been injected with jellyfish gene. They took a glowing gene, injected this glowing gene in this rat and made it glow under ultraviolet light. So as you can see, it's green.
So I'm getting a red one in November. Green one in November. And you can have them also. The blue isn't very bright, so there's no impetus to try and make it blue. Yeah. So it's not easy to make the first one. Use fertile. So if I have male and female, we had it for a while. And you get this honey jelly beans to make love. But she ate them. So. Is he blind? No. He's not.
So see, it's glowing here. This because of genes. Genes can change anything or add any new features to the animals, to the plants, or to anything that we want to add. Okay. Naeem, are you there? Fatima? Fatima, can you get the mic, please?
Hello, good morning.
Good morning. How are you?
Hi, Miss Hanan, how are you doing?
Alhamdulillah, I'm fine. Okay,
Sorry
so
for being late. Just we
no,
had generator.
it's okay. It's okay. So we were talking about adding genes to animals or to plants to increase to improve the offsprings. Okay, so they can take genes, new genes, they can extract it, they can inject it. This is called genetic engineering. So genetic engineering is adding new genes to cells. Okay, we all know that when during reproduction, okay, the nucleus can copy itself, should copy itself before splitting or before dividing, okay? So how can this happen, or how this can affect the genes, or how the genes can be copied? The engineered mouse egg cell were fertilized with normal sperm. As we saw here, we have a glowing rat, right? Or glowing mouse. What they did, they took this engineered mouse, which is the glowing one, okay? And they took egg cell from it and they fertilized it with normal sperm without any glowing genes and began to multiply. When they add these together, they kept splitting in two to form all the cells in each embryo. Each new cell had a copy of the glowing gene. Okay, let's see this together. If we said this is the cell, okay, and here we have a nucleus, right? This is the egg cell, and this is the nucleus. Then a sperm came in, diffused with the nucleus, with its head, and the nucleus, both nuclei diffused together. This egg cell has the gene, the new gene for the glowing. And sperm cell, normal. It was normal without any adding genes. Then when they fertilized these normal sperm with the nucleus of the new gene, they became fertilized and they start to divide. In reproduction, we said that the nucleus should copy itself, right? Because it needs to copy the genes. So each fertilized egg now has what? Naeem. Each fertilized egg has what exactly?
A normal sperm cell.
No. With a copy of what?
Fertilized egg. Just no. Copy of a new gene.
Yeah, with a copy of the new gene. Okay. When this fertilized egg start to split in more than one egg, okay, what will happen now? What we will get? Let's say it start to split, okay, in two cells. What we will get here?
Identical twins?
No, not identical twins. they are starting to divide to have more rats, right? To have more cells and become an embryo, right? So this cell, and this is another cell,
Okay.
right? Each cell,
It's one cell, then it became
it become more cells until it will form the embryo. So the rat now, the new rat or the baby rat or the embryo rat, it has a copy of the new genes in all of its cells, right?
Yes.
Okay. So it takes skill, the engineered mouse eggs, egg cell, we said, fertilized with the normal one. Then it began to multiply, okay? When it starts to split or keep splitting, all the genes, all the cells, they will have a copy from the new gene, okay? So the cells in the glowing mice contain gene from jellyfish. The jellyfish genes has the glowing gene. The jellyfish body has the glowing genes. So when they injected this, the rat became glowing, start to glow because it has this gene. When it has offsprings or babies, are they going to have the same gene or without this gene?
I think different genes.
Why? Are they going to
If,
have the jellyfish gene, the glowing gene, or not?
of course, they will
They
have
will
glowing.
have.
What?
Because like they contain genes from the jellyfish, yes?
Okay, this is about the mother, okay?
Yeah, yeah. So mice will have like different genes from jellyfish and other things.
Okay, I mean, are they going to pass these genes to their kids or not?
They will pass them.
What, Sophie? Sorry?
Yes, because it will be genetic.
They will pass them to their offspring.
They will pass them to their offsprings. Fatima, are you there?
Yeah,
Can you answer?
yeah, I think it will pass because
They will
it will be
pass.
genetic.
This will be genetic. This will be in their genes. Since the fertilized egg, we are not talking about the egg cell, non-fertilized. No, the fertilized egg. Okay, now it became a cell. Okay, it will start to multiply. Since it has the glowing gene, it will pass it to the offspring. So they will get this gene. They will have this gene. Okay, this is about copying gene. Okay, how can they grow medicines using genetic engineering? We all know that some people, some scientists, they need to use genetic engineering to grow medicines or to make medicines. It takes skill and hard work to make new genes to work in animals or plants. Bacteria are easier to use, their cells are simpler. Guys, we all know that cells have very simple units, right? Can you tell me anything about the cell, the bacteria cell? Is it like, does it have, do they have many cells or one cell or two cells?
I
Guys,
think they
anyone?
use it. I think it's useful.
No, I mean like the bacteria itself.
You mean the job of the bacteria?
No, no, no. I mean the bacteria cell. Are they multi-cells or unicell or what?
Oh,
They're unicells, right?
Yeah, unicells, okay? Unicells, they have only one cell, okay? They don't have nucleus in their bodies and their genes are loose in the cytoplasm. You know, they look like long strands. Let's say this is a bacteria cell, okay? They have very long strand like this, okay? And they have circular ones They call them plasmids. They call them plasmids. They don't have nucleus. Their genes lies in these strands, okay? Guys? Okay. So the bacteria has no nucleus and the genes are loose in the cytoplasm. You can find them in this long strand. They also reproduce very rapidly. Can you tell me anything about reproduction? How can they reproduce themselves? By what?
Reproduce. You mean the reason why they like reproduce
No,
very fast?
how can they reproduce? By splitting, right? Splitting themselves in two parts. Keep splitting, like dividing in more than two, or more than one. The one cell became 2, then became 4, then became 8, became 12, like this, splitting. That's why they are reproducing very rapidly and easily. They are splitting themselves, like dividing their bodies in their cells in more than one, okay? We all know that the diabetic people suffering from lack of insulin, right? What is this insulin? Guys, can you be active, please? We are about to losing time. We are losing time.
Insulin, I know, like it's insulin. Kind of injection.
Okay, they are doing what? What the function of this injection?
You know, the way I know it, like people use it when they get poisoned.
No, not poison. When they have diabetes, high blood sugar, right?
Yes.
Yeah, I always
High blood
Oh,
glucose.
yes, they use
This is called diabetes, okay? Your body normally excrete insulin from the pancreas. Your pancreas is producing this insulin to keep your blood sugar or blood glucose stable, not high, not low, okay? So if you are eating too much chocolate, for example, your pancreas will excrete this in your blood, so it will decrease the blood glucose, so you will not be diabetic, okay? So some people, they have high blood glucose, which is diabetes. Their body cannot produce, or they cannot make insulin, enough insulin to decrease the blood glucose. So they have to get injections, okay? It's not easy to get injections. So how scientists are using genetic engineering? They are using the cell of a bacteria. Why? Because bacteria is easy to multiply, easy to reproduce, okay? So it will be easy for them to extract or to get more insulin in short time, okay? So since the bacteria have no nucleus on their genes, they can inject the human insulin inside the cell. So the bacteria with the insulin gene produce a human insulin for these people to inject themselves. I'm going to show you a video how they are doing this exactly and how they can extract this insulin from it, okay?
Excuse me, Ms. Hanan.
Yeah.
Can like a human remove the pancreas or no?
They can, but this will give them like troubles. If the pancreas is damaged, they have to remove it. But this will cause many problems, okay? Okay, look at this one. This is the bacteria cell, okay, on your right. This is the bacteria cell. You can find, you can see the strands, right? You can see the strands.
Yes. Yeah.
Okay, you have here this one, okay, the circular one. This is called plasmid. You can find the bacteria genes in it because it's hard to get this one, and like divided and add genes to it. So it will be easier to use this one, okay? The circular plasmid. So they are taking this plasmid, cut it like this, because it has this section, it has genes, okay? They need to cut it and add another gene to it to complete the plasmid, and in this case, the insulin gene to it, okay? So we will see how they are doing this exactly. Need to inject this in the human, in the bacteria genes. Guys, can you see the video?
You
No.
You cannot see it?
stop sharing screen.
What?
You stop sharing the screen.
Okay. Can you see it now?
Yes, now you can
Yes.
see it.
Okay.
Can you like?
Let's see together, okay? So as we said, on your right, this is the bacteria cell, and on your left, these are the genes for insulin. See, we have a half circle here, okay, almost circle. This is the plasmid, okay, and they got the blue one, which is the insulin gene from humans, and they need to join them together.
So this way helps the pancreas to work well.
No, it's not about the pancreas. No, it's not about the pancreas. We
Oh,
are talking
only
about they cannot make insulin in their pancreas, so we need to give them injections. That's how they are making the insulin, manufactured the insulin, like the artificial insulin, or not artificial, sorry, manufacturing or reproducing more insulin outside of the human's body. They're using the bacteria because they are easy to multiply. That's why. So it will be easier for them and cheaper for them. See, now we have complete plasmid. So the gene that enables the production of human insulin and the bacterium plasmid combine to form the recombinant plasmid. This is like the mixed plasmid, human insulin with the bacteria's plasmid together. In this case, they will inject it back to in the bacteria. Now, see, it became to split. The recombinant plasmid is transferred to the bacterium cell, and bacteria are enabled to multiply. When they start to multiply, each bacteria, each new cell has a copy, see? They have the same plasmids, the blue and the orange one. All of them, because they are copying their genes, keep copying their genes and multiply.
For the blue one,
Yeah.
is it a human cell?
Human insulin, DNA, gene from the DNA.
Ah, I thought it was bacteria too.
What?
I thought it was for the bacteria.
No, they took the insulin gene from the human, okay, from human pancreas, okay? Then they extract the gene injected in the plasmid.
How do they know which gene is for which?
What?
How do they know which gene is which gene is for which gene is the insulin gene?
Each gene has its own structure, has its own codes, because genes they are like codes. So they can know, they can test and extract the genes that they want exactly. These are like codes, not just like mixed or they can see. Genetic engineer, they can they are special in genes only. They study genes, they are studying these codes, so they can know the insulin code. They can extract it from the pancreas itself take the insulin and take the DNA from the insulin and take the genes from this DNA. Okay?
Okay.
So this is how can they grow the insulin. Transferring genes to bacteria. Most of the genes in bacteria are in one big and the rest are in little loops called plasmid. That's what we were seeing now. It's easier to insert new genes into plasmid. The process involves 3 main steps. As we said, these long strands, one big strand, okay, it's complicated to add genes to it, but these loops or these plasmids, it's easy to cut a gene from a human insulin and add it to the plasmid to continue to complete the loop. So how can they do this? They have 3 main steps. First, they need to identify and extract the gene you want to use. It's, for example, like this insulin, okay? They need to take insulin, they need to extract the genes from it, from the DNA, okay? That's what they want to use. Then they need to insert the gene into the plasmid, injected or added to the plasmid, which is the loop that you saw in the video, okay? Third thing, they need to add the plasmid to bacteria again, okay? It's like giving, you know the artificial babies? That they are taking the sperm cell and the egg cell out of the body and fertilize them together and injected them back to the mother's.
Oh, yeah.
It's like this one. They need to identify the genes, insert it inside the plasmid and give it back to the bacteria. The bacteria will start to multiply. So each cell will contain what?
Each cell
Gene.
plasmid will contain an insulin cell. Insulin gene.
Yeah, copy of the new genes, which is the insulin or whatever we want from the bacteria to make, okay? Also, they are using genetic engineering in farming, okay? How can they do this? Using genetically engineered plants and animals to produce medicines. Sometimes they can make this in the cow's bodies, Let's go to the next page. The insulin gene has been added to cow and goat embryos. When the animal grow up, they will make insulin in their milk. Of course, this milk is not for drinking, They need to extract or separate this insulin out because some people with the diabetes, they don't just need to drink milk or added insulin to milk with added insulin to get better. No, they need to inject it in their blood. Because why? Milk is going to the stomach, right? So how can they get enough oxygen, enough insulin immediately? They need it for injection. So what they are doing, they are adding the insulin to the cow or goat embryos. The embryo, when the embryo grow up, have milk, they will have the gene in their milk. They need to extract the insulin from the milk and make injections. Of course, this process is kind of hard. Why hard? It's not even convenient for manufacturing insulin because this is taking time. Okay, you need to wait for a long time until the embryo form, until the cow deliver the baby or the offspring, until the offspring become an adult to extract milk. And this is taking too long, okay? So bacteria is the easiest way because they can multiply rapidly so they can extract the insulin from them easily. Also, yes?
Does any milk we drink or any milk contains insulin?
No, this is added insulin. This is
only the animals that has been added to them insulin.
Yeah, only these ones. Yeah, but your body is not gonna take a benefit from this insulin too much. It needs to be injected in your blood so it can decrease the blood glucose. because your pancreas is extracting this in your blood. Okay, artificial spiders. They make artificial, they call them artificial spiders. We all know about the spider silks. You know the spider silk, right?
Yeah.
Oh, okay, silk.
Yeah, when you touch it, how does it feel?
Sticky.
And
Yeah, something like that.
right?
Like it contains something. It contains something like liquid.
Yeah, it's like elastic and strong. You cannot break it easily. It will just keep
Yes.
like, sticking in your fingers. It's not easy to break. Spider silk could make ultra light fabrics. They're so light, but they are strong and elastic. Spider silk is far better than manufactured silk. So if people need this silk from the spider, they need to add this silk making gene, okay, to other organisms. So how can they do this? They cannot get enough from the spiders, the natural spiders. So scientists put their silk making genes in specific kind of bacteria. You know why? Because bacteria
So they produce enough amount of the silk.
yeah, why? Because bacteria is easy to multiply, so they can have this genes, so they can extract it. What the problem is, not all bacteria can hold this gene because it's like a poison for them. They can get poisoned and they can be killed by or destroyed by this gene. So we have specific kind of bacteria that can hold the spider silk gene or making gene. It's called Salmonella, okay? This Salmonella can hold it and it can make it, it can survive also. So after multiplying, it will throw the silk out of the cell. As you know, it's like a foreign material. It will throw it out as fast as they make it because it's something foreign, for something weird on inside their bodies. Try to imagine like bacteria is making silk inside their bodies. Okay, so they need to take it out of their bodies. They are throwing out this silk after making it. Okay, so this is how can they?
Hanan,
Yes.
Salma said she couldn't join.
Okay.
It said it's locked. The meeting is locked. She's trying.
It's not locked.
I don't know, that's what she told me.
It's okay. Anyway, we finished. Okay.
Miss, can you go up again, the writings? I want to do a screenshot because in the beginning of the lesson, I had some problems. I couldn't
Moving
write.
genes, this one?
Yes.
Okay. Guys, if you have any question, because I need to stop recording. It's time.
Mm-hmm. Okay. Okay.
I'm just taking a photo.
What?
Yeah, can you go? Hello? Miss, can you go to the next page?
Okay.
Yeah, okay, everything is clear now.
Okay, guys.
Yeah.
See you next time. Okay. And don't forget the yellow box. Don't forget the yellow box, please.
Okay.
Yeah, okay.
Bye bye.
Have a good day.
Goodbye.
Thank you.